Okay, good afternoon, everyone. This is the final stretch. Thank you all for staying with us. I think it's been a lot of interesting and exciting conversations. As you have probably noticed throughout the day, we have actually brought the voices of all relevant stakeholders in our Ladder Cures Accelerator ecosystem. We've had of course, academic, leaders like Jonathan Weisman, for example, who gave an amazing lecture this morning. And then, we had Jimmy Olageri, who gave a fantastic talk from the patient perspective, a sickle cell disease patient who scaled Kilimanjaro. So, remarkable story. And so now we're hearing from Fiona Marshall, who actually represents for us another very important stakeholder in this ecosystem that we're all a part of. And that's, of course, the biopharma component. And that will be a theme for our conversation during the panel discussion that David Meeker will lead a little later this afternoon. So I think Fiona Marshall needs no introduction, but I will go ahead and introduce her. She is the president of the biomedical research at Novartis. The job that a senior faculty member once told me is the most coveted job in Kendall Square. So she actually there leads more than 5,000 scientists in 6 research sites dedicated to the discovery of biomedicines across the Novartis ecosystem and across the entire pipeline, leading basically R&D development for Novartis. Prior to this position, Fiona served as Senior Vice President and Global Head of Discovery and Early Development at Merck. And prior to that, she was the founder and CSO of Heptarcus Therapeutics, a UK-based company that was focused on structure-based drug design. So she has a rich background from which to draw for all the discussions that we've been having today. It's really a great honor to have you here today, Fiona, so thank you very much for coming.
Thanks so much for inviting me, Anna, and thanks for everybody being here on a Friday afternoon. So it's a privilege to come to the Broad, and just such a wonderful institute that I've obviously followed for many, many years, actually, since its inception. So I'm going to talk to you about some of the work we've been doing at Novartis, but particularly tapping into some of the themes of this meeting in rare disease. Obviously, as a company, we work very broadly across many, many different diseases, ranging from some of the very most common through to very rare diseases. This is really just a sort of overview of the company. So we have focus in 4 main therapeutic areas. Cardiovascular, renal and metabolic fall under one therapeutic construct. Oncology, immunology, and neuroscience. Now, we don't have a separate rare diseases unit, which some companies have, although that changes in the industry quite a bit. But what we do certainly work in rare diseases, but within these core disease areas. And then we do certainly try and combine expertise on our platform technologies and share that expertise and knowledge across the different disease areas. One of the fortunate things of working for Novartis is we do a lot of different modalities, probably more than any of the other pharma companies. So as well as small molecules and biologics, we do have gene therapy, xRNA as we call it, which can be many different types of oligonucleotide therapy, as well as gene and cell therapy. And we are a global company, one of the largest biopharma companies. And really for us, we are trying to deliver high-value medicines, also to as many people as possible. So as well as, and thinking really about the highest unmet need. We also do a lot of work in the global health area. And I was really pleased that we won the award for the highest access to patients last year. So Novartis medicines reached 300 million people around the world, and that included over 30 million who were in low to middle income countries. So we do try and enable access whenever we can. One of the things I'm sure we'll get to talk about on the panel is sort of alluded to on the final column. I mean, there are a lot of headwinds in terms of execution of delivering new medicines. I would say it is getting harder. I've worked in the industry a long time. And we are continually trying to challenge ourselves to improve R&D productivity. And from a research point of view, choosing the right targets and making sure those are the right targets for the diseases that we want to treat is really the way that I think about how to improve productivity. It's really removing all those, or certainly reducing the numbers of failures that we have. So this really summarizes what we're doing in biomedical research. So yes, I am very lucky to have this job. I would agree with that part of it. We do work across these incredibly important and interesting and challenging disease areas. So I have the disease area heads. We also have a group called DAX, which is supposed to be sort of any disease area that we think there could be a breakthrough. And in fact, interestingly, that's where we have our renal portfolio now, a growing one, which I am going to tell you a bit more about. And then a new group which I've set up since joining Novartis called DARE, which is diseases of aging and regenerative medicine. And that's thinking still about these same diseases, but in an orthogonal way. Why is it that aging contributes to so many different diseases, and what are those pathways? And we'll probably talk about nodal biology later, but I do think that aging and certain parts of the aging process really are key nodes in diseases of aging that we can target. So again, we work across many different technology platforms, and I'm going to describe some of these in a bit more detail, and particularly the ones that I think are most relevant to the rare diseases space. Hence this slide. Okay, so let's start with gene therapy, AAV gene therapy. So we were fortunate enough to be able to bring Zolgensma, AAV9 gene therapy to market. When we acquired Avexis and we first started working on AAV9, of course, we did think it was going to have a lot of opportunity in many different diseases. And as we already heard from our previous speaker, that hasn't been as easy as we first hoped. One of it is, of course, the immunity that some people have to AAV, but also the tropism of those capsids, especially to CNS neurons, was actually much poorer than we first expected. So we've put a lot of effort also into engineering new capsids, both internally but through additional business development. We work with Voyager, who many of you may know, and we recently acquired a company, Kate Therapeutics, and they very much focused on the muscular dystrophies, so really evolving myotropic capsids that can deliver genes to both muscle, skeletal muscle, as well as cardiac muscle. I'm not going to say too much more about that, but happy to talk about that in the questions. One of the modalities I'm going to focus on today is siRNA. I think this, for me, is one of the most exciting modalities for the treatment of a wide range of diseases. I think the next generation now is not just about delivering siRNA to the liver, but it is about more targeted delivery to specific cell types. And we've been doing a lot of work in that area to really try and tie down different opportunities across, again, both rare as well as common diseases. Now, coming from a structural biology background, I still really like small molecules. Oral small molecules are a drug that you really can easily deliver to patients around the world. But what's fun now is all of the incredible extra knowledge that we have from structural biology through techniques like cryo-electron microscopy, but also now predictions like AlphaFold, really allow us to design chemical compounds that can change protein function either to increase or decrease or alter expression levels or alter protein-protein interactions in very unique and novel ways that are really allowing us to target pathways that were not before possible. And a sort of an extension of that is also the targeted protein degradation, where you can really essentially knock out a protein by causing its degradation. And again, a project that I didn't have time to talk about today, but I'd like to, is on our sickle cell disease, where we are using a degrader of a transcription factor that actually converts adult and fetal hemoglobin, and that's now in clinical trials for sickle cell disease. But again, we focused on that as a modality by thinking about who are the patients that need this drug. So one of the considerations on choosing a modality, one is what problem are you trying to solve, but also who needs that drug. And of course, all of the patients, we actually have sickle cell disease as a part of our global health portfolio, and we think that treating those patients with an oral small molecule is the one that will give the most access. So I'm going to say a little bit about what we're doing in the genetics field, because this is another area that I've grown significantly since joining Novartis, being a real believer in the power of human genetics to inform on target selection, not just, again, for rare monogenic diseases, but also much broader diseases. I mean, there really, I don't think, is a disease that doesn't have a genetic component, and we're still really learning that the more we look and dive into the data. But one of the areas that we're doing is really to set up a new group. Some of you may know Jeremy Jenkins. He's the head of our Target ID group. Within Target ID, we have a genetics group. And really the objective here is to try and find new targets, test out new hypotheses, and interrogate those in different ways to try and prioritize our selection for drug discovery. We work very much matrix now across the organization, so very close interactions between the research and the development and our commercial colleagues. We do have to take them their view into account. Unfortunately, there are some rare diseases which are too small for us to work on, but we do work really as a group together, thinking about what is the high patient unmet need, what are areas where we can really add value and work on together. A lot of this comes down to data. One of the things which is very exciting now is the quantity of data that's available in genetics. And I'm old enough to have been there around at the sequencing of the human genome, all the expectations that we had in 10 years' time, everybody was going to be treated with and have all their DNA sequenced. Well, that didn't happen in 2010. We're now 25 years later, but I do feel that we are really getting there in terms of the amount of data that allows us to get these incredible associations and genetic information. But the data is still not in all the right format, and we're still not very good at asking the right questions. So where do we use genetics now? Actually, it's across many of the things that we do. And it's not so much about those monogenic disorders, although we are working on those, but it's really about those genes that alter your risk, either increase or decrease your risk of broader diseases that we're working on. So we're trying to identify targets, target validation, from databases and genetic cohorts. We're also using it for biomarker discovery because, again, I think in terms of clinical trials, ideally we want to really accelerate clinical trials by having much better biomarker readouts, another thing we could pick up on the panel. Also use genetics to enrich trial design and also use genetics to really understand about target safety. And there's a number of different ways in which we're doing that. But overall, what I really want to do is try and shrink that timeline. I mean, a timeline that you showed of how long it takes to get those, and those should have been fast timelines in a disease like muscular dystrophy. Our timelines really need to be short, much shorter, half the time. I mean, not 10 years, 5 years. And we need to do everything we can to make that happen. So again, one of the big things for me is getting all of the data that we can. And human genetics data is one of it, of course. Single cell data, single cell profiling data is another. Getting all these multimodal data that can come together is what gives you that confidence to move forward in a target. I'm not going to go through all of these different data sources that we've invested in. They all have different benefits in terms of the types of patient populations, the sizes of the data, the sizes of the population, but also whether we have access to health records, to exome sequencing data, to samples, also whether there's an opportunity to recall the patients when we make interesting observations, and also whether you can also then trace back the family history is also incredibly useful. And I'm glad to say that there's a lot of public data and public initiatives and public-private partnerships where we work together, particularly with other pharma companies, on these types of programs. So again, I've got a few slides, keep going back to the same problem. It is the data. Is the data in the right place? Is it in the right architecture? How can we search across different data sources and compile these? One of the things we've done is a big effort, which we call Data 42. It's to construct a data lake of all of our clinical trials data over essentially the last 20 to 30 years of Novartis. So we have huge clinical trials database, and in many cases with all of the health records, biomarkers, what trial were the people in, how did they respond, were there any safety issues. So this is incredibly helpful for when we go and try and understand new problems. If a safety issue comes up, we can see whether it's happened before, and we can do our own sort of studies, reversing information from clinical trials back into the research portfolio. But as you know, those of you, I'm sure everybody here, what we're looking for are these strong genetic associations and trying to understand whether mutations, what is their functional consequence, and doing a lot of work on that in silico, which is informed by things like AlphaFold, where you can map mutations onto a protein and predict it, as well as actually, of course, doing the wet lab work, which at the end of the day is something you always do have to do. More complicated is all of these mutations that are in the non-coding regions. What are they doing? How do you understand their function? So it's really about closing that gap between these genetic associations and the mechanism. And well, this is a really great example, which I haven't sort of put a slide on, but I probably should have done, SLC30A8. So this is a zinc transporter protein, and a GWAS study was published to say that this was associated with your risk of getting type 2 diabetes. But there wasn't, it was literally at that sort of level, just a whole load of genes associated with type 2 diabetes. Working with collaborators in other pharma companies, as well as the hospitals in Pakistan, 100,000 people in Pakistan, we have their whole exome sequencing. We're able to look and see, were there any people who had mutations in this protein? And actually within that cohort, there were 15 human knockouts of this. And that ranged from homozygote total knockouts through to heterozygotes and different degrees of loss of function. And we then were able to call those people into the hospitals and study their glucose levels as well as do glucose tolerance tests. Turned out if you had a loss of function of this protein, zinc transporter protein, you had reduced basal glucose levels, but in the glucose tolerance test, you were much better at mounting an insulin response. So this really helped to validate that initial GWAS study in humans, both from the point of view this could be a good target in diabetes, but also it's safe. There are human knockouts, perfectly healthy, walking around. So that really gives us confidence to go forward in a target like that. And beautiful when you can actually get this gene-dependent correlation with function, which we saw in this case. So I'm now going to give two little snapshots of a few things that we're doing. First of all, in the neuroscience area, and then in the kidney area, in honor of Anna. I thought that would be a good one to pick. So starting off in the neuromuscular diseases, and I think I'm trained in neuroscience. These are the ones that I think, especially in children, are some of the most heartbreaking diseases and ones that really can either be fatal or cause lifelong disability. So the disease I'm just going to tell you about is Charcot-Marie-Tooth disease, CMT1A disease. And this is actually relatively common. It's one of the most common inherited neuromuscular disorders. And it's due to not a mutation, but actually a gene duplication in this protein PMP22, which is a myelin protein. And it's interesting, there are diseases also with this same protein where you get a loss of function, but either gain of function or loss of function results in abnormal myelin. And so then you get abnormal axonal impulses, delayed conduction. And in these people, what happens is in early childhood or adolescence, this is really most important in peripheral nerves. You get tingling and then loss of proper muscular tone and function in arms, wrists, and in legs. So then it causes problems with walking and trying to be involved in sport. And the disease does progress over the lifetime. So one of the ways where, given that this is a gene duplication, of course, what you want to do is knock down the gene, but back to normal levels. You don't want to knock it out completely. And this is much easier to get the titration of a knockdown with an siRNA than it would be with a gene therapy. So this is a technology, but we need to deliver that siRNA to the right tissue. It's not the liver. We need to deliver it to the motor neurons. And so this is a company we acquired, DTX, based in San Diego. They had developed libraries of lipids which could be conjugated to siRNA, and then those different lipids would actually be taken up preferentially into different cell types. And you could select out the lipid conjugates for different cell types, but also liver D-targeted. And we call this the Falcon platform, fatty acid lipid conjugated. An example here isn't from the CMT1 program, but this is actually for delivering to the CNS tau proteins. This is for Alzheimer's disease. And you can see really nice knockdown of tau right across different neurons in the brain. So going back to Charcot-Marie-Tooth, again, further validation. We've got the genetics, which is pretty clear. But let's just check if silencing PMP22, or at least knocking it down in a mouse model of CMT1, will give you the expected functional benefit that you would be looking for. This also helps guide what you want to measure in the clinical studies. So what you've got here is a mouse model. It has an overexpression of PMP22, so pretty similar to the human disease. Has the same sort of phenotype. It has it from birth, starts to develop muscle weakness, and you can look there. I don't know if this has a It probably doesn't have a pointer, but anyway, you can see that those sections are through the nerve fibers. So you've got the wild type, big thick myelin, insulation around, and then in the disease, a loss of the myelin, but also then axonal loss as well. And then down in the bottom right-hand corner, so that's the treatment with the siRNA for PMP22, you get remyelination or reduction in loss of myelination and axonal protection. This improves nerve function. And it also, most importantly, improves locomotor function, in this case in the mice by beam walking or grip strength. And these same measures, all of these, you can use in the clinical study. So this is the candidate molecule that we've designed, EDK060. And so this is an siRNA to PMP22 with a lipid conjugate that delivers it to neuromuscular tissue. And again, this is now taking that exact molecule into an animal model, but now you've got to put in the human gene. So this is overexpressing human PMP21. And you can see exactly pretty much similar data to what I showed you in the last experiment. Reversal of the loss of myelination, axonal loss, really nice. Again, this nice dose-dependent dose titration, which is going to be very important when we go into the clinic. Again, dose-related improvement in nerve conduction and in grip strength. So this is at the point where we literally are about to go into the clinic and we'll be measuring all of these same endpoints. Okay, so let's switch now to kidney diseases and iptacopan, which is a molecule which we've now brought forward as a drug to the clinic. Fabhalta is its name. So the node here, if I'll call it that, is the complement pathway. Complement pathway is, I think, one of the most interesting and complicated pathways, really fundamental to our immune system, our innate immune system. 50 different proteins, all very carefully coordinated and in a cascade. And of course, this coordination of all these proteins, a lot can go wrong. If you have mutations in these that alter this carefully coordinated process, then you are going to have complement-mediated problems. Maybe you don't mount a proper immune response and you're susceptible to infection, or maybe you have an overactivation of the complement pathway. And so a whole range of genetics associates different polymorphisms, deficiencies in the whole range of complement genes in a whole range of disorders. Now, there's multiple complement pathways, but the one we're most interested in is the alternate pathway, known as AP for short. And the drug that we've been working on, iptacopan, you can see there, works as an inhibitor of factor B. That's part of the C3 convertase, so it forms the enzyme that creates the downstream cascade. And ideally, I think in nodes, Anna, you probably agree, you want to hit up at the top of a pathway because then you can cover everything down below it. So that's why we think in this case hitting factor B is a good idea rather than going down to the things at the bottom. And the alternative pathway genes are associated with a range of diseases here, macular degeneration, number of different kidney diseases, and other hemolytic disorders. So just focusing in on what is the evidence for the alternate pathway, complement pathway, in these two kidney diseases. First disease on the right, this is C3, which stands for complement, so it probably is related to the complement pathway, C3 glomerulopathy. Now, these patients, many of them do have direct genetics of overactivation of the complement system, but some of them have acquired the disease through autoantibodies that are activating the complement pathway. But basically, when you do a kidney biopsy, what you see is a whole load of accumulation of complement proteins in the glomerulus. They basically act to block up the filter. And so you get an accumulation of protein and proteinuria, and that triggers then kidney damage. Another disease, which I'll say more a bit about later, but I just want to show you again the complement here. This is different. It's not driven by complement. That isn't the driver of the disease from the genetic point of view. In fact, in this case, interestingly, the genetics shows certain complement polymorphisms are protective in the disease. It turns out that complement is downstream of this disease. The actual cause of it is to do with, it's an autoimmune disease. But then you get complement activation that adds to the disease. But nevertheless, complement is still the green accumulating in the glomerula, and patients have elevated complement proteins, especially the downstream ones showing complement activation. So LNP023, this is what iptacopan was called while it was still in the lab. This is an inhibitor, very nice inhibitor, again, optimized by structure-based design, oral molecule, very potent. And this is now a model in rats. It's a general model of glomerular nephritis. What you do is you actually take renal extract. I looked up this model carefully so I could understand it before I came to tell you about it. You take extracts of the kidney, inject it into rabbits, and you get a polyclonal antibody response. When you inject that back in, of course, those antibodies basically go straight and block up, again, the glomerulus, cause a lot of damage, very similar to those human diseases. And so you get proteinuria increasing over time after this anti-FX1A injection. What you can see in the blue lines is this is now treating with iptacopan, really reducing the proteinuria. And what's interesting is it will actually work whether you give it after the antibody has been given, so after the disease is ongoing, but it can also be preventative and prophylactic. Now that's important when we start to think about when would this drug work. So normally you'll start in clinical trials in later stage disease, but those people have already lost a lot of their kidney function. So ideally you want to move it into early disease as quickly as possible and stop people losing their kidney function. So now going into the clinical trial, this is in the C3G, and the panel on the left, I won't go into too much detail, but this basically confirms that C3G is definitely due to activation of the alternative pathway. You can distinguish that through EGTA and EDTA, but it also shows that LMP023 can really normalize all of these complement fragments within the serum, and most importantly then produce this really nice reduction in proteinuria over time, actually 45% reduction over this 80-day trial. So then going back to IgAN, and this is really the most common form of primary glomerulonephritis. Now what happens here is that certain people, for some reason, and there is some genetics here, produce galactose-deficient antibodies, galactose-deficient IgA. So they're not glycosylating those antibodies properly, but the antibodies are fine, they're fine, and you could go around with that and you wouldn't even get the disease. But then certain people who have those, some of those people also have certain aspects of their immune system, maybe it's HLA related or whatever, will form autoantibodies to those galactose-deficient IgAs. Now you've got a horrible complex of the galactose-deficient IgA with now IgG, big complexes in your blood, again, blocking up your kidney filtration system, causing a lot of kidney damage. Now you get complement coming in. And so then the complement is adding to those complexes, causing more inflammatory mediators to come in and really causing a lot of damage within the kidney. So if you can block, there's different stages here. You could go for a B-cell therapy to try and stop those autoantibodies, but you could also try and stop the complement as also really another way of delaying the progression of the disease. And you can see here again in the phase 2 study a really nice impact on proteinuria and also really completely blocking these complement. This is the terminal part of the complement pathway in these patients. So finish up with this slide. It can happen. We can get drugs for rare diseases all the way through to market. And we were really pleased that we actually this year, only a few weeks ago, got the first approval for Fabhalta, as it's now called, as an oral treatment for C3G. And also, we also previously got approval for IgAN in the US. And now, of course, we're trying to roll this out around the world. But make sure these patients are also getting diagnosed, especially as early as possible, because a lot of kidney disease can continue before you actually get the proper diagnosis. So getting that urine test done on a routine basis is really important. Okay, thank you.
Thank you. That was amazing. Thank you, Fiona. Questions for Fiona before maybe in the background we can put the chairs out. Thanks, Katie. Any questions for Fiona? If no one else is asking a question, I will ask a question while we're putting the chairs up. So I think this, thank you for being a kidney example, but I think it is like a very recent success.
It is, yeah.
And I think it really speaks to the fact that, we've been talking about all day today about the idea of, well-defined causal biology, which of course often based on genetics, as you said, is a way to understand mechanisms, and some of our colleagues here brought wonderful examples from all kinds of different fields and diseases. But here's an example in which it sort of plays out, right? Like you understand maybe a more genetically defined, more rare entity like C3G. But then actually, interestingly, and this is the story that I wanted to understand better from a development perspective, so we can all sort of understand from you how that works. How is it that it was approved in IgA nephropathy before C3G, and how did you manage the development of the drug from this rare, more defined entity into something that's more common? Clearly it had to do with your preclinical development path, but I'm curious if you can say more about it.
Obviously, we were doing both in parallel, in fact, because we knew that there was already that strong complement biology, so it made sense to go and one of the things we actually are doing in pharma now is we used to just do one disease at a time, and then if it worked in that disease, then we'd do the next, then we'd do the next. Well, we don't have time to do that now, and so we try and do everything in parallel, which is more risky, but if it works, then you've got, all of those indications right from the beginning. So we did start both at the same time, but, IgAN is a more common disease, and so it was easier, then it becomes easier to recruit the patients, and so, and then, then you're still having the discussion with the regulators on what are the endpoints. As you know, you've got proteinuria. Are you going to get approval just on that? Is there going to be any effect on renal function through GFR? But of course, that's a very long trial in these diseases. They are slowly progressing diseases as well. So, that's another consideration. But, luckily, and I think it makes total sense because we know that proteinuria is a direct measure of renal function. So we did get approval based on that.
Fabulous. Oh, Raj, you have a question? You can ask while we're assembling is that okay, assemble our panel? So I'll ask Wendy and John and David to come up. If you have a question, you need to get the mic.
Please ask. Yeah, please do.
Casual. We're trying to be very easy going here.
Thanks for the great talk. I was wondering your opinion about whether or not academic labs should engage in the drug discovery and development process. Here, we're good at target discovery for rare disease, and we publish the paper. But if it doesn't immediately catch fire, then we start to think about should we pursue finding a therapy? I don't know if you think that's insurmountable.
No, I don't think it is insurmountable, and I have seen where that has been done. Reasonably successful. Well, not reasonably, where drugs have come to market just from academic institutes. That has happened. Particularly in the cancer area, actually. So, and I think in some of the modalities are getting easier for academic labs to do. I would argue an siRNA is probably easier than trying to do all the chemistry and toxicology and all of that. So it is doable, but of course you need the funds from somewhere, and you need to have the right people brought in with the right knowledge. But it certainly is doable, yes.
You can ask a question. You're popular, so one more question. Go for it.
Hi.
You mentioned that one of the applications of human genetic data could be better trial design.
Yes.
Could you perhaps elaborate on that? Also, in the context of rare diseases, how could human genetic data improve?
Yeah, so as I say, now we know with all the GWAS studies, we know that people with particular, mutations or polymorphisms may be better responders to drugs or not. So making a decision about which genes you're going to actually have sequenced as you enroll patients. Now, you can either do it where everyone comes in the trial, but then you afterwards work out what all the different genetics are, or you can select. And of course, in the case of the rarer diseases, let's say Huntington's disease is another disease we're working on at the moment. We want to understand, what the number of repeats, exactly what their status is from a genetic point of view, because that actually alters the rate of progression of the disease as well. So it is very important to understand the status. And, even when we have a drug that could alter it's mutation independent, if you like, it's still very important to capture that information and then see separately how patients with different mutations will progress.